Hello and welcome to the last part of chapter 16, the sense organs, light and vision. So chapter 16, we discussed equilibrium, we discussed hearing, we discussed cutaneous stimulation, these proprioceptors, mechanoreceptors on your skin, sensing the touch, on your ear, on the tympanic membrane, sensing the mechanical vibration. Now it's time to discuss photoreception, vision. How does light get into your brain? Because remember, we discussed that the brain has the roof, the cortex, and it's has different centers. It's center for balance, center for hearing, center for vision. So today we're going to talk about how is vision being processed into your brain. So let's begin by defining vision or sight. Vision is your ability to understand what you're seeing. So the mere ability of visualizing things is not enough. You have to understand what you're looking at, which involves your brain. So your eyes are connected to your brain in a direct sensory visual projection pathway that does not involve the spinal cord. And what do we see? We see objects because they either emit light or light is reflected off of them. So if you're seeing something, you're seeing it either because that something you're looking at is emitting light, or it's because that something you're looking at is reflecting light that's shining on it. So vision is the perception of objects in the environment by means of the light they emit, as you see here, or the light that is being reflected. Light is a visible electromagnetic radiation. So we do not see the entire electromagnetic spectrum. We only see from 400 nanometers wavelength, and that is represented by the ultraviolet radiation. That's the minimal wavelength we are able to perceive, the 400 nanometers ultraviolet radiation. And the maximal wavelength that we are able to detect on the electromagnetic spectrum is the 700. So we see from 400 ultraviolet radiation up to 700, and that is infrared. So from the name, it's kind of a little bit counterintuitive. How come the 400 is less than 700? So why is it the case that the 400 nanometers represent ultraviolet, while the 700, the big one, is representing infrared? Shouldn't the infra be 400 and the ultra be 700? No, because the 400 nanometers is reflecting a short wavelength, but definitely the ultraviolet radiation has more energy. So that spectrum is reversed. You have less wavelength, but high energy. So 400 nanometers, ultraviolet radiation has low wavelength, but very high energy. That's why we call it ultraviolet radiation, ultra energy. While the 700 nanometers, that represents a high wavelength, but very short energy. That's why we call it infrared. So here the human vision is limited to the wavelength from 400 to 700. We discussed that. The light must cause photochemical reaction to produce a nerve signal. So there's a transduction. We have a photoreceptor that's going to be sending photons, light energy that will be transduced and converted from light energy into electrical energy. Ultraviolet radiation less than 400 nanometers has too much energy, and infrared has more than 700 nanometers. Okay, we begin by discussing the accessory structures of the orbit. The orbit is this socket that contains the eye. So the orbital region is the area around the orbit, the eye socket, that contains structures that protect and aid the eye. You have the eyebrows. The eyebrows is there to enhance your facial expressions and also to block any sweat that's coming down to your eyes. The eyelids, also known as the palpebra, they are consisting of the orbicularis oculi muscle, and the muscle around your oral cavity is known as the orbicularis oris, but the muscle around your eye is known as the orbicularis oculi. And it has a supportive tarsal plate. It is covered with skin and it has tarsal glands. What do these tarsal glands secrete? They secrete oil that is going to reduce and prevent the tear evaporation. We don't want tears to evaporate because we want to keep the eye structure moist. What is the conjunctiva? The conjunctiva is a layer that is covering the white of the eye and the inside of the eyelid. Do not forget that part. Conjunctiva is not only on your eye, it's the eye and the inside of the eyelid. That's why when you get itchy eyes, your eyes hurt and the inside of the eyelids hurt also. Get itchy, red, inflamed, swelling, pain, redness, heat. So these are all signs of the inflammation of the conjunctiva, known as conjunctivitis. This transparent mucous membrane lining the eyelids and covering the interior eyeball, except for the cornea. That cornea does not have conjunctiva on it. That makes it a good key point for purposes of examination. Tell you which part of your eye does not have the conjunctiva, and the answer is the cornea. And the conjunctiva secretes thin mucus film to prevent a drying of the eyeball. It is richly richly innervated and heals quickly. The lacrimal apparatus, it's a gland and a tear duct for the production and the drainage of the tears. Lacrimal is for secreting the tears. Tears have lysozymes. These are bactericidal enzymes that are there to become antibacterial. These are macrobicidal enzymes, and they're there to lubricate the eye and deliver oxygen and nutrients and prevent infection with bactericidal lysozymes. There are tear ducts in the lacrimal apparatus, and let's not forget also that there is a cushion around the orbit of the eye, and that is known as the orbital fat. It protects the vessels and the nerves that are in the eye. So this is a very nice figure that shows us the main structures of the eye. You have this lacrimal caruncle, and this is where the tears are being secreted. The palpebral fissure is this big fissure that opens up, dividing the top eyelids from the bottom eyelid, known as the orbital fissure. And this big sulcus here, this big groove, is known as the superior palpebral sulcus, while the bottom one here is known as the inferior palpebral sulcus. And the two edges represent laterally. So you know from our anatomical position, the more you're moving toward the center, that's medial, and the more you're moving to the outer regions, that is the lateral region. So that's the lateral commissure, and you also have the medial. The opposite to it is the medial commissure. So let's now look at the structure of the eye. That's the bone, the frontal bone, and if you look here, the eye is in this socket known as the orbit. In the orbit, you have very important muscles. You have the levator palpebrae superioris muscle and the orbicularis oculi. That's the one that fits the socket. And you have the cornea. That's the most anterior part of the eye. And right behind it is your conjunctiva. So there is no conjunctiva on the cornea. This is the lacrimal gland that secretes the tears into the eyes. And you know the tears escape into the nose, the nasal cavity, and sometimes the tears are swallowed into your esophagus. Okay. There are very important muscles that you need to know these muscles, and you need to know the nerves that affect these muscles. The extrinsic muscles of the eye attach to the exterior surface of the eye. That's why they call them exterior external muscles. And the external muscles of the eye are the superior, the inferior, the medial, and the lateral rectus. These are the extrinsic eye muscles that attach to the extrinsic surface of the eye. In addition to that, you have the superior and inferior oblique. So from the name, superior, inferior, medial, lateral, rectus. Rectus means that these muscles are structured in a straight line, while the superior and inferior oblique muscles, they are in a curved fashion. They are structurally curved, and these help with the transverse movement of the eyeball. That's why if people have any inflammation or defect or damage to the oblique muscles, you see they will have to rotate their entire head just to be able to move their eyeball in an oblique manner. Why? Because the oblique muscle is damaged, is terminally damaged. You need to know the nerve. So the superior and inferior oblique muscles, they are innervated so there's also a very important fibrous ring, fibrous structure. The superior oblique tendon passes through this fibrous ring called the trochlea. And that's a very good area, a key concept for purposes of examination. They will ask you on the exam, what is the name of the ring? The fibrous ring is called the trochlea. This fibrous ring, what's the name of the muscle that goes right inside it, right through it? And the answer is that's the tendon of the superior oblique muscle. So the cranial nerve 4 innervates the superior oblique. The cranial nerve 5 innervates the lateral rectus. And the cranial nerve 3, known as the oculomotor nerve, innervates all the remaining other 4 extrinsic muscles. So that shows you the superior, the inferior, the medial, lateral rectus muscle. They move the eye up, down. So the superior rectus moves the eye up. Inferior rectus moves the eye down. Medial rectus moves the eye medially, and lateral rectus moves the eye laterally in straight fashion, like a cross, right? East, west, north, south, up, down, right, or left. These are your rectus muscles. However, the inferior and oblique muscles, they turn the 12 o'clock pole of each eye toward or away from the nose. So they either gonna shift your eye towards the nose transversely, obliquely, in a curved fashion, or away from the nose. So they produce the slight elevation and depression of the eye. So that shows you, that's the superior oblique. And you see that's the fibrous ring, known as the trochlea. And this is the inferior oblique muscle. And these are your lateral rectus. On the other side, the medial rectus. And then you have the superior rectus and then the inferior rectus. And that's cranial nerve number 4. The trochlear nerve is going to be influencing the superior oblique muscle, while the abducens nerve, cranial nerve number 6, is going to be affecting the lateral rectus muscle. And all of the other remaining muscles, they're going to be innervated by the oculomotor muscles. So that's a very good key concept to understand for purposes of examination. They will ask you, what is the name of the nerve that is responsible for the movement of the superior oblique muscle? And that's the trochlear nerve. They can ask it the other way around. The trochlear nerve is going to move which muscle? And as we said, the trochlear ring is the fibrous ring in which the superior oblique passes. So that makes sense. The trochlear nerve moves the superior oblique while the abducens nerve, the trochlear nerve is cranial nerve number 4, while the abducens nerve is cranial nerve number 6, and all of the abducens nerve is moving the lateral rectus muscle. It moves the eye towards the lateral component, and all of the remaining muscles are moved by the oculomotor muscle. Okay, so now if we want to know the structure of the eye, the structure of the eye is that the structure of the eye has 3 main components. The first component is the tunics, and those 3 tunics or those 3 layers are either fibrous layers. There's the first outermost layer, the outer shield, the outer case is the fibrous layer. Right beneath it, we're going from outermost to the innermost layer. The outermost layer of the eye is known as the fibrous layer. The second most layer of the eye, right below the fibrous layer, is known as the vascular layer. It's like a grape. When you remove the skin of the grape, don't you see like another layer? So that's known as the uvea. That's the middle layer, and it has the choroid and the ciliary body and the iris. We will talk about that some more. And then you have finally the deepest most layer, deeper to the uvea or the vascular layer, is the nervous layer or the retina and the optic nerve. The fibrous layer is the outer layer derived from 2 regions. You have the sclera, which is the whole white part that is covered by the conjunctiva. And then you have the cornea, which is the anterior transparent region that admits light into the eye. The vascular layer or the uvea is the middle layer with 3 regions. The choroid, it's a highly vascular and darkly pigmented layer behind the retina. And you have the ciliary body, which is an extension of the choroid muscular ring around the lens, and it supports the lens in the iris and secretes aqueous humor. The iris is a color diaphragm that controls the size of the pupil. So it's because people who have dark eyes is because there's a lot of melanin in the chromatophores of the iris. The inner layer is the nervous layer, the retina, and the optic nerve that is able to take the photo stimulation and convey it and transduce it back into the brain. So the retina is actually part of the brain. The retina is a visible part of the brain that you can see non-invasively. You do not have to, if you want to see your brain, you have to crack open the skull. But there's another way to see a part of your brain without invasively performing a surgery or opening the brain and getting to see what's inside of it, just by looking through an ophthalmoscope. If you see the retina, that is your brain. The retina is a visible part of your brain. But if you want to see something else besides the retina, then you need to open the skull. The only visible part of your brain that is visible without invasion or without surgery is definitely your retina, the retina. So that's the sclera. That's the outer fibrous layer. And it does not encircle the eye all around, because once we get to the interior part of the eye, you have the cornea. Then the second layer is the uvea or the vascular layer, and it has the choroid. And after the choroid, there's a ciliary body that's going to be secreting the aqueous humor into the anterior compartment. And you have the lens. The lens is going to stretch vertically, become so thin, or it's going to expand horizontally. And we will see the differences. And then you have this optic nerve that's going to take all the stimulation and send it to the brain. Now, let's talk about the optical components of the eye. The optical components of the eye are transparent elements that admit light, and they refract the light rays and focus the images on the retina. You have the cornea. The cornea is a transparent window allowing light into the eye. So all of the optical components of the eye, they are a transparent elements that admit light and they refract the light rays and they focus images on the retina. The cornea is a transparent window allowing the light into the eye. The aqueous humor is a serous fluid secreted by the ciliary body into the posterior chamber between the iris and the lens. It flows through the pupil to an anterior chamber between the iris and the cornea, and they are reabsorbed by the scleral venous sinus at the same rate that is secreted. The optical components of the eye are, you have the lens. The lens is a transparent structure composed of flattened, compressed lens fibers, and they are suspended by suspensory ligaments from the ciliary body. And the pull of the ligament changes the shape of the lens to help focus the light. The lens are rounded with no tension from the ligaments, and they are flattened with the pull of the suspensory ligaments. The vitreous body is a jelly that fills the vitreous chamber behind the lens. This jelly maintains the intraocular pressure. It helps hold the retina against the wall of the eye and the blood supply. The hyaloid canal is an oblique channel and a remnant of the artery in the embryo. Production and reabsorption of the aqueous humor. So we see that the ciliary body or the ciliary process is going to be secreting this fluid. And you see the suspensory ligament, and that's the lens. Cataracts represent clouding of the lenses, and the lens fibers darken with age. And, this is the clouding of the lenses. There are fluid-filled bubbles and clefts filled with debris appear between the fibers. They are induced by diabetes, smoking, drugs, ultraviolet radiation, and certain viruses. Treat by replacing natural lens with a plastic one. Because once the lens is damaged, it is damaged beyond salvage. It is damaged irreparably. What is glaucoma? Glaucoma is pressure within the eye due to the obstruction of the scleral venous sinus and the improper drainage of the aqueous humor. So we're unable to drain the aqueous humor. There's an imbalance. So aqueous humor is being secreted at a high rate more than it's being absorbed. The pressure within the eye due to the obstruction of the scleral venous sinus and the improper drainage of the aqueous humor. The death of the retinal cells are due to the compression of the blood vessels and the lack of the oxygen. Macular degeneration is the death of the receptor cells in the macula, which is the central part of the retina. And that's going to lead to loss of the vision in the center of the visual field, and it cannot be cured, but the progression can be slowed. Diabetic neuropathy is a retinal degeneration caused by the effects of diabetes mellitus on the retinal blood vessels. It causes most adult blindness in the United States, and it can often be prevented with early detection and control of the diabetes. You see, that's cataracts, and you see, the glaucoma is, if you have glaucoma, that's how you're going to be eventually able to see. You'll only see the central part of the eye. The neural component, these are the last layer or the innermost layer. The retina forms from an outgrowth of the diencephalon, known as the optic vesicle. They are attached to the eye at the optic disc, which is the posterior exit of the optic nerve, and the ora serrata, which is the anterior edge of the retina. The retina is pressed against the rear of the eyeball by the vitreous humor. And the detached retina causes blurry areas of vision and blindness. The retina exam with the ophthalmoscope shows the fundus or the rear of the eye. The macula lutea. Lutea is the spot. Lutea is yellow, and macula is the spot. It means the yellow spot, which is a 3 millimeter patch of cells posterior to the lens along the visual axis. The fovea centralis is a pit in the center of the macula lutea, which produces the most finely detailed images, and the blood vessels of the it contains the blood vessels of the retina. So here we see the arterioles and the venules, and the fovea centralis is the central location, and the macula lutea is the yellow spot, which is a 3 millimeter patch of cells posterior to the lens along the visual axis. Okay, this blind spot is a very important concept because the optic disc is occupying a location in which there are no photoreceptors. So because there are no photoreceptors, there is no detection of light. So that is going to become blind in your visual field. However, your blind spot is not necessarily a big blotch. It's not a big black blotch spot. Why is that? Because your brain evolved something known as visual filling. So your brain is going to fill that big blotch, that big spot, with what it assumes would be in that location. So for example, if you see this really long green bar and you're looking at this X location and focusing on this X location, and you start to move the paper without looking anywhere else except at the X. So imagine you're just looking at the X and this slide starts to move to the left or it moves very right. If it moves far to the left, there will be a point where you will not see the red dot. You will not see the red dot because that would be your blind spot. But because you cannot see the red spot, what are you going to see? Are you going to see a big black blotch? No, you will see a green, even though there's really no green thing here. But your brain is going to do visual filling. It's going to fill the red spot with that green bar because it's going to assume it's better than to have an empty or black blotch or a blind spot is to put that extension of green, even though there is no green, but it's better to put that green than nothing. That is called visual filling. Okay, the visual process represents light that enters the eye and it focuses on the retina and produces a tiny inverted image. The iris diameter is controlled by two sets of contractile elements. There's the pupillary constrictor, which is a smooth muscle encircling the pupil. The parasympathetic stimulation is going to narrow the pupil, and the pupillary dilator is a spoke-like myoepithelial cell, and there's sympathetic stimulation that widens the pupil. The pupillary constriction, so you have to understand that the pupil, if it widens, it is because the pupillary dilator has been contracted. And if it constricts, it's because the pupillary constrictor muscle contracted. So both are contraction. This is really important. Both are contracting. If your pupil constrict, who constricted it? The contraction of the pupillary constrictor. And if your pupil dilate, why did it dilate? Because The pupillary dilator contract. So it's contraction, contraction. So why are we dilating the pupil? Why are we contracting the pupil? We're dilating the pupil to accommodate more light and more visual stimulation. So you can tell that's important for survival. If you're being chased by a lion, you have to be able to see as much as you can so you can survive. So you will see in sympathetic nervous stimulation in fight or flight, your eyes are going to dilate. They're not going to constrict. But when you're all relaxed and you're all comfy and happy, you don't need that much visual stimulation, so your eyes are going to constrict. What dilates the pupil of the eye? The constriction of the pupillary constrictor muscles. What makes the pupil dilate? The contraction of the pupillary dilator muscles. What's the photopupillary reflex? The photopupillary reflex is the pupillary constriction in response to light. When so much light is jumping at you, now that is like a stressful stimuli. So much light is coming into my eye. That's really stressful. So what does it make sense? For your pupil to dilate, you have so much light coming into your eye. What is better for your survival? For your pupils to dilate or for your pupil to constrict to protect you from all that so much light coming into your eye? Obviously, to constrict. So it's a protective, compensatory survival reflex. The photopupillary reflex is a pupillary constriction in response to light. What is refraction? So now we're going to talk about the formation of an image. What is refraction? Refraction is the bending of the light rays due to the refractive index. So it depends on the substance. Something that's very special about light compared to sound. Sound does not travel in vacuum. Nobody makes a sound in space because space is vacuum. Sound travels in solids, sound travels in liquids, sound travels in air. Sound does not travel in vacuum. Unlike light, light does travel in vacuum. Space is vacuum. Do you not see light in space? You do, because light travels in vacuum. Now, vacuum is going to have a refractive index that's different from gas. Vacuum is going to have a refractive index that is less than gas. Gas is going to have a refractive index that is less than liquids. Liquids are going to have a refractive index that is less than solids. So if you shine light in vacuum, it's going to be different than shining light in air. It's going to be different than shining light in water. It's going to be different than shining light in glass. So they're all going to bend the light differently. The thicker you are, the higher the refraction, the higher the refractive index. So if your refractive index is low, you bend the light just a little bit. If your refractive index is high, you're going to bend the light really, really so much. There you go. So if we look here, the lens has a refractive index, and we need it to do so because we need to focus the light into the retina. Imagine you had no lens. Light is going to be scattering all the way in the back of the eye, but you need the lens so it can break and refract the light into the back of your retina. The near response, also known as emetropia. The near response, so emetropia is when the eye is relaxed and focused on an object that is more than 20 feet away. So the light rays coming from the object are essentially parallel. So the rays are focused on the retina, are going to be focused on the retina without effort. The near response is an adjustment to close range vision. It involves three processes to focus the image on the retina. So there are convergence of the eyes and constriction of the pupil, and that leads to accommodation of the lens. Okay, conversion of the eyes, convergence of the eyes. So eyes orient their visual axis toward the object. So that is known as the conversion of the eyes. The constriction of the pupil known as meiosis, which is different from meiosis, meiosis. This is meiosis. It's the constriction of the pupil. It blocks the peripheral light rays and reduces the spherical aberration known as the blurry edges. Accommodation of the lens, that's going to represent the change in the curvature of the lens, and the ciliary muscle is going to contract, and the ciliary suspensory ligaments are going to slack, and the lens is going to take a more convex, thicker shape. So convex shape looks like this. Concave shape looks like that. Convex, concave, convex, concave. So there is a near point of vision. That's the closest an object can be and still come into focus. And it lengthens with age. So if we look here, emetropia is when your vision is not converging, meaning that your pupils are not centered on one point. If you look at convergence, that's a property where you focus both eyes into one point. You make like a triangle. But in emetropia is when you're all relaxed, you're not focusing on anything, so you're not using convergence. So you can tell if you're reading a book, you're using convergence, you're making a triangle. But if you're just out in the woods and you're just seeing, you're just looking at the forest. Now you're able to see. Now you're able to see in a emetropia. So emetropia is when you are in a relaxed, non-convergent state. But convergence is when you are focusing both eyes into a particular point. So this shows emetropia, so the light is being focused in the fovea. But when there is convergence, there's pupillary meiosis, and the lens is going to perform accommodation. So we're going to focus the light even at more sharper point. If we're going to look at something far, that's known as emetropia. So we're going to be pulling the lens vertically, making the lens so thin. But if we want to see something that's really close, we move the lens horizontally, making the lens wide. This is really important. What happens to the lens if you want to see something far? What happens to the lens if you want to see something close? You need to know that. You need to know that if something is really far, your lens is gonna become so thin. But if you want to see something that's so close, the lens is gonna become so wide. So if we now look at, sometimes if we're looking at a particular object and it is so close to us, if it is so close, the light rays are going to be crossing in the back of the retina. So we're not going to see them properly. That's why we use this convex lens, and the convex lens is gonna make the crossing. You see, before the lens up here, uncorrected, there was no lens. So they cross in the back of the lens. But when you wear glasses, convex lens, what happened to this extra space? It went a little bit back. So it shortened it. So the convex lens, it does not diffract the light. It brings together the light and closer. So now we have corrected the vision. So if you look at this person, you can tell, oh, this person is looking at something so close. This person is looking at something that is, oops, you're looking at something that is so far and it's crossing in the back of the lens, in the back of the retina. But you correct hyperopia, farsightedness with a convex lens. They will ask you that. They will ask you, if you look at something like that, what is this person suffering from? You can tell, oh, since the light rays are crossing in the back of the retina, that means they're looking at something really far. So we need to correct it with a lens that shortens and brings the light towards back toward. We don't want any extra light to the back. We want to cross it on the retina, and the convex lens is going to do the job. So farsightedness is known as hyperopia, is corrected with a convex lens. While if you're looking at something that is so close to you, now that's the opposite now. In farsightedness, the light rays cross in the back behind the retina. But in nearsightedness, they cross in front of the retina. So both are problems. One time you're crossing behind the retina, one time you're crossing in front of the retina. You don't want either. You want it to be crossing on the retina, like here, like emetropia. In emetropia, you're crossing on the retina. So in order somebody that has nearsightedness, you correct it with a concave lens. And this concave lens is going to spread out the light, going to diffract the light, push out the light more towards the back, give it an extra push. You know, when you push the car, you push extra from this line, you push this line back. But the convex is going to pull the light back. So one is going to pull the light back to the retina, one is going to push the light to the retina. Or you need to sit down and look at this some more. It's very important to know that. The cellular layout of the retina. You have the pigmented layer. That is the most posterior part of the retina. It absorbs the stray light, so the visual image is not degraded. The neural components of the retina from the rear forward. So the photoreceptor cells absorb the light and generate a chemical or electrical signal. But they are not neurons. You have the rods, the cones, and certain ganglion cells. So only the rods and the cones produce visual images. Bipolar cells. So bipolar cells are going to communicate between the photoreceptor cells and the ganglion cell. If we look at, this is amazing, but this is the actual scene. This is an actual histological image. That's how exactly your eye look from the back. But it's not going to make sense unless we look at a diagram. You know, a diagram is like a model. The model can sometimes help you understand more than the actual image. But this is the model that's going to help you understand. That's the back of the eye. So the back of the eye has the photoreceptors. It's pretty interesting. The photoreceptors are not out front. The photoreceptors are out in the back. So when somebody comes into your house, they enter through the front door, and then they go into the living room. And then if you have something in the backyard, then they go through the kitchen, then they go into the backyard. But imagine you have a guest that comes from the backyard, then out into the kitchen, then to the living room. Then this is how light does. Light does not get detected from the front. It gets detected from the back. So light does enter through the door. It doesn't enter from the back. Light doesn't enter from the back of your skull. It enters through your eye, but it doesn't get detected at the frontier. It gets detected all the way in the back and moves backward. It gets detected here in the back. So you must recognize you have 3 layers. layer number one. Can everybody see this is layer number one? That's layer number two in the middle, and that's layer number three. Layer number one is the photoreceptors. You have the rods and the cones. Those are the photoreceptors. So you need to know where are the rods, where are the cones, all the way in the back. These are my photoreceptors. Rods detect darkness, light in the darkness, while cones with the letter C, like color, C for color, C for cone. So cone detect colored light. Then the photoreceptors are going to move from the light from the first layer into the second layer known as the bipolar cells. And the bipolar cells are going to pass the light into the ganglion cells. And the ganglion cells, you know, when you hit ganglia, you're talking about a nerve now. So now which nerve do we deal with in the light and vision is the optic nerve. So, you know, the ganglions are going to be sending the information to the optic nerve. So it's a little bit counterintuitive how light gets detected. Light enters through the eye, but gets detected in the back of the eye. Then it gets sent forward to the photoreceptors. Then the bipolar cell, then lastly the ganglion cell. Okay, so rods are used for night vision, known as scotopic vision, and cones are used for photopic or color trichromatic vision. This is the structure of a rod. That's the structure of the cone. And visual pigments are very important to understand because rods contain visual pigments known as rhodopsin. And I wish that they called it the retinal opsin. That would have made more sense. But they didn't call it the retinal opsin. They call it rhodopsin, but rod is a retinol. So retinol, that's why they say eat carrot. It's good for your vision. They mean it's a big important part of rod in the rhodopsin. So deficiency in vitamin A leads to a depletion in the retinine. And retinine is part of the rhodopsin, so you're not going to be able to see well at night. So that's going to cause night blindness. The cones do not have rhodopsin. Cones have photopsin. Photopsin. So that shows you that's the disc. And so in rods, the light changes rhodopsin. In dark, retinol is bent. So these are the reactions that take place in the structure of the rod. And this is cis retinol when there is no light. But once there is light, the natural structure of retinol is to be in the cis formation. But once there is no light, it becomes bleached, and that is known as the trans retinol. So in dark, the retinol is bent, known as cis retinol, and retinol and opsin are together. When light shines, it straightens retinol, it becomes trans retinol, and retinol is going to dissociate from opsin. This is a process known as bleaching. You see, now they're going to separate. To reset, it takes 5 minutes to enzymatically that's why the rods are not activated in the light. So in the light, they are separated from one another. They don't work. But in the darkness, so that's why when you turn on the light in the middle of the night, it takes a while for you to be able to see well, because that is about the time it takes for trans retinol to become cis retinol. That's the time it takes for the rods and the opsin and the retinol who are being separated, and now they're going to be reattached again. They're going to be reattached together again, and the retinol and opsin are going to be stuck together, and that's going to make them enable you to be able to see. The cones function in a similar manner, but it doesn't have, like we said, retinol and opsin. It has photopsin that regenerates a lot faster than rods and cones. So that's the procedure. Cis-retinol is because opsin and retinol are attached. Once you shine the light, they're going to be inactive. They're going to be in a state of cis-retinol is going to isomerize to trans-retinol. So opsin is going to trigger a cascade that breaks down cyclic GAMP and the cessation of the dark current. The signal is going to be created in the optic nerve. And the trans-retinol is going to separate from opsin, and it's going to then be regenerated back into opsin. Okay. This is very important because we see that if there is darkness, rods are going to be secreting glutamate, and glutamate is going to be performing as an inhibitory neurotransmitter that's going to inhibit the bipolar cells from secreting any neurotransmitters and preventing any stimulation to the ganglion cells and the optic nerve. But once there is light, the glutamate secretion is going to stop, and bipolar cells are no longer being inhibited by glutamate, and bipolar cells are going to release a transmitter, and that's going to signal that is going to be signaling a light stimulation. Scotopic versus photopic vision, that's very important because here you see that there is a lot of convergence. So in scotopic vision, night vision, you see multiple rods converging onto one gang of bipolar cells and multiple bipolar cells converging into one ganglion cells. But in photopic vision, colored vision, you see a more specialized one-to-one ratio. We have one cone fusing into one bipolar cell, fusing into one ganglion cell, and then sending the information to the optic nerve. And that is what we need. In photopic vision, there is more color detail, so we need more specificity. So there's one-to-one ratio. One cone, there's no convergence. Unlike in that at night, you don't need that much specificity. At night, you just need the rods to help you not hit the table or hit the wall. So you just have multiple rods fusing into one bipolar cell and multiple bipolar cells converging into one ganglion. But here you don't have that. You have one for one. You have one cone fusing into one bipolar cell, fusing into one ganglion cell. Duplicity theory says that rods are sensitive and respond in very dim light, and you need a system for the resolution, and you need a system for coloring. So in color blindness, people who are colorblind, looking at this particular image, the problem is with red-green light. So they will never be able to see this number 74. They will just see all of these dots, but they cannot tell that this is there's a 74 in here. But a person with no red-green color blindness, they will be able to see this number 74. Stereoscopic vision is our ability to have the eyes sharing a visual field, part of the visual field. Unlike horses and rodents that have their eyes on the sides of their heads, so their vision is like a panorama. That's a panoramic vision, not a stereoscopic vision. So a horse can see one side of the visual field, and the left eye will see the other side. Both eyes of the horse or both eyes of the rodent can never have one shared visual field. But we can, you see here, that's an example of a shared visual field. Like here, you see this green light this green region is the region that is shared by both eyes, while yellow and blue are unique. So the yellow part is unique to the right eye, the blue part is unique to the left eye, while the green part is shared by both. Okay, so light is going to be Interpreting the occipital in the occipital lobe. The last thing you need to know is that the left eye has an ipsilateral fiber tract that sends visual stimulation to the left primary visual cortex. So the left visual cortex receives visual stimulation from where? From the left eye and from the left side of the right eye. They both converge into the left visual cortex, while the right visual cortex receives visual stimulation from ipsilaterally from this right eye and from the right side of the left eye. Okay.